Good morning, I am Madhurima Datta, Zoology and Biology Educational Content Creator. Now's content for B.Sc Zoology Honours, Microbiology Honours, Physiology Honours and M.Sc in Zoology, Microbiology and Physiology. Paper Genetics and unit is the Introduction of Genetics and topic of Introduction of Genetics, Functional Gene and Non-Functional Genes. This video for also CSNET Life Sciences students, CUT PG student, GATE students and DBT students and also IIT JAM aspirant students. What is the genetics? Genetics is the branch of biology that studies heredity. What is the heredity? How physical or biological traits are passed from parents to offspring. And genetic variation, that is why individuals within the same species look and functionally different. Next, it is the increasing pace of genetic discoveries has become staggering. The Human Genome Project is a case of point. This project began in the United States in 1990, when the National Institute of Health and Department of Energy joined forces with international partners to decipher the massive amount of information contained in our genome, the deoxyribonucleic acid found within all of our chromosomes. Next, the hierarchy of genetic information. To understand how genetics work, it helps to see how genetic instructions are stored inside living organisms. At first, we know about the cell. It is the fundamental unit of life. Most human cells contain a nucleus that acts as command center storing genetic instructions. Next, chromosome. Thread-like structure housed in nucleus. Humans have 23 pairs receiving one set from each parent. Next, DNA or deoxyribonucleic acids. The chemical molecules that make up chromosome and DNA forms a twisting double helix structures containing 4 chemical bases, that is adenine, thymine, cytosine and guanine. Next, it is the most important question, what is the gene? It is the hereditary factor which is responsible for trait. It is the unit of information encodes genetic characters. It is a particular encoded sequence for genetic character. And genes is 2 types, that is the functional gene and non-functional gene. Next, as scientists have attempted to unravel the mysterious within our genes, this journey have involved the invention of many new technologies. An example in human recombinant insulin sold under the brand name Humulin, which is synthesized in strains of Escherichia coli bacteria or E. coli bacteria that have been genetically altered by the addition of genes that encode the functional region of human insulin. Next, the molecular expression of genes. First, I will discuss about the trait. What is the traits? In genetics, a trait is any specific characteristics or features of an organism. It's essentially any identifiable quality from the color of your eyes to how efficiently your body digests milk. Traits are part of your phenotype. Phenotype means the observable physical or functional expression of your genes. And phenotype is determined by your genotype. What is the genotype? The actual genetic code stored in your DNA. Next, how traits work. Traits are generally classified into 2 main categories based on how they originate. At first, inherited traits. It is characteristic passed down directly from parents through DNA via genes. Example, natural hair color, blood type, attached versus unattached earlobes. Next, acquired traits. Characteristics developed during our lifetime as a result of environmental influences or physical practice, which cannot be genetically passed on. That is, callus on finger, scars or muscles built through the weightlifting. Next, think of DNA as a matter instruction manual located inside the cell. A gene is a specific section of the DNA that acts as a recipe. That is, the cell needs the gene to produce a protein, and the protein performs a job, builds a structure which creates the final observable trait. Now the sequence is gene, then they produce protein, and protein give the result of the trait. Next, we know that the genes are two types, that is the functional genes and non-functional genes. At first, I will discuss about the functional genes. A functional gene is an active segment of DNA containing the complete instruction needed to synthesize a functional product. And how it is works? It includes essential regulatory regions, that is like promoter region, that tell the cell when and where to read it, and followed by intact coding sequences, that is exons, that translate into protein or functional RNA like RNA or rRNA. Next, role. They drive all living processes, building cellular structure, producing enzymes, carrying out metabolism, and maintaining organ functions. Examples of functional genes are HBB genes. This genes provides instruction for making beta globin, a crucial part of hemoglobin, the protein carrying oxygen in red blood cells. HBB genes full form is hemoglobin beta genes. Next, INS genes or insulin stimulating gene encodes the insulin hormone responsible for regulating blood sugar levels. Next, non-functional genes. A non-functional gene, often referred to as a pseudogene, is a DNA sequence that resembles a normal gene but has lost its ability to code for a work protein or functional RNA product. How it happens? Over time, evolutionary mutations accumulate in the sequence. This can include premature stop codons, frame shift mutation, and promoter mutation. This topic I will discuss in elaborate in the next slide. Next, the short note of the premature stop codon, or what is the premature stop codon? It is a trailing or early signals that cut off protein synthesis midway. And what is the frame shift mutation? Insertion or deletions of letter in the DNA sequences through of the reading frame. Next, promoter mutation, inactivation of the on-switch region, preventing the cell from initiating transcription. Next, role of the non-functional gene. They often remain dormant in the genome as evolutionary fossils, leftover traces of genes that were once active in our ancestors but are no longer needed or useful. Next, example of the non-functional genes, chylopseudogenes or L-gulonolactone oxidase. In most mammals, this gene actively produces vitamin C internally. In human and other primates, mutations deactivated these genes long ago, making us dependent on dietary vitamin C, that is scurvy risk, while still leaving behind the inactive pseudogene sequence in our DNA. Now the topic has ended. For all video of genetics in B.Sc Zoology Honours, Microbiology Honours, Physiology Honours and also M.Sc, please subscribe my YouTube channel Biogenesis by Madhurima and follow my Facebook account Madhurima Datta.